I'm Regis. My name is Bill Litster. I'm the business development manager for the West Coast. And I'm not going to talk up here about Regis, but I am going to give you the 1-minute commercial speech. I have to do that. So, I mean, a lot of you know what we do at Regis. We are an API, strictly small molecule API manufacturer, as you well know, and we're centrally located in Chicago. We have some commercial products, so we are FDA inspected. That's important to note. But we are a strictly small molecule API manufacturer located just outside of Chicago. Have excellent process chemistry. We work on a lot of early stage projects from preclinical, phase 1 and beyond. Sometimes the majority of our projects are the earlier stage where we have process chemists that you transfer your process in. Process chemists are excellent. Take a look at that and make tweaks on the process. And we have an excellent analytical group that's fully equipped with equipment and support with the GMP synthesis. The analytical group has got everything. We have everything in-house. We outsource just a few things: bioburden, XRPD, and high sophisticated particle sizing. But other than that, we have all the equipment in-house. Some of you have maybe been there, but the best thing to do is to visit our company when you get serious and want to really know what we're about. And we also have other unique capabilities: chiral separation group. We have an excellent chromatography group. Unfortunately, but fortunately, we have a lot of chromatography experience with processes. So again, and another unique thing about us is we are family owned. We're one of the few, I think we're the last standing small molecule API companies that hasn't got bought out or merged. The reason I joined there a lot is because I felt like they didn't want to do that, and there's a benefit to that. We're small and flexible, and we can handle projects. We don't want to get bought out, as far as I know now. The family doesn't want to do that, and they've been around for over 60 years now, the Glunz family. So Louis Glunz is the president. His father is the owner of the company, or founded the company. Unfortunately, Louis, he wanted to be out here, but he has a couple little kids at home. He couldn't travel, so he left it up to us. So that's kind of us in a nutshell. If you have any other questions, talk to me after the presentation, or Andy Miles here. I'm going to introduce Andy Miles as our business development director for Regis. Supports the Midwest as well, and that's pretty much it. So that I don't want to get too much into the detail of Regis, but that's just kind of a quick elevator speech on what we are. So I think you're all here to hear Steve, not me. So I'm going to introduce Steve, and I'm just a real quick introduction. I don't know much about you, but I have got some little cheat notes here, and I pronounce your name correctly, hopefully. Dr. Steve Baertschi, and Steve runs Baertschi Consultants in Indianapolis. So where you're from in Indianapolis, and you worked at Eli Lilly for over 20 years. 26. Now that's all I know. So if you want to get into I've heard a lot of good things about you, and I had some of my clients say that you're an excellent speaker and they enjoy your talk. So I'm going to leave it up to you. So hopefully enjoy and you'll learn some things. And again, Steve will be available after his talk to answer questions if you have time.
Okay, without any clock in here, I'll try to maintain. The trouble with new Apple Watches and phones is they go blank, right? And so then you can't just glance down. So you see me coming to tap, and I'm just trying to make sure I don't keep you too long. It's good to be here. Thanks to Regis for inviting me. I've been to Regis headquarters. That's how this kind of happened. They invited me up, and I did not know much about Regis outside. Are you hearing me okay? I didn't know much about Regis outside of HPLC columns and chiral columns, which is what they've been known for many, many years, as long as I've been around. HPLC, that is. And so it was good to get to see their facility and realize that there's a lot more to Regis than just HPLC columns and that kind of expertise. I'm going to talk today a little bit about impurity control strategies, and for some of you, really for anybody, that is a really potentially huge field, because you could have a 2-3 day workshop easily and fill it up on impurity control strategies, especially if you're focusing around impurity control strategies within process development, which is where the real control for the API is all around the process. But I'm going to touch on that a little bit, but focus more on degradation-related impurities, partially because that's my expertise, and second, I believe that it's more well-developed than the degradation. I believe that the process impurity control strategies are more well-developed than degradation-related control strategies. And we're going to see whether or not this works okay. If I do, got to go the right way. Okay, so I'll give you a little introduction. You've already heard a little bit about me. I'm going to just briefly in the introduction, I'll go through and I'm going to describe what control strategy really is, maybe from a definition, definitionally, why, which is sort of obvious. Put it into context of development timeline and the fallout rate, the reality of fallout rate in pharmaceutical development, and then go into a little bit more about mutagenic control strategies, because mutagenic impurities are the new challenge that's out there to develop control strategies at such low levels. Describe briefly a case study, because most of what I'm going to talk about is not a case study. So most of it's going to be framing the issue and giving you some data and getting you to think about the topic for a while, and then obviously make some conclusions. What I'm going to cover in today's talk is going to be organic impurities. I'm not going to cover residual solvents or elemental impurities or extraneous contaminants or extractables, leachables. Those are going to be separate topics that could be their own workshop. I'm just going to focus on both ordinary, so sort of ICH Q3AB type impurities, as well as mutagenic, which is ICH M7 type impurities. And I'm going to try to convey the idea that understanding something about mechanism, chemistry, can guide your control strategy better than just empirically doing statistical studies. So the take-home message, I'll start with the take-home message, is that impurity control strategies evolve over the development timeline, and that's because there is a fallout rate to the development of new molecules. And if you utilize that fallout rate and think about a fit-for-purpose approach, which means you can kind of cheat a little bit. You're not doing a marketed product type control strategies all the way through. It can really dramatically reduce the resources you need. That impurity control strategies are best developed with a chemistry-oriented scientific approach versus a technique-oriented. A technique-oriented would be throw analytical techniques at it, throw statistics at it, and see what you come up with, and without developing a scientific understanding. That impurity control strategies are more complicated and challenging for mutagenic impurities than they are for ordinary impurities. So the regulatory landscape around impurity control strategies kind of starts with ICH Q1A, which is a stability and photostability guidance, Q1A-B, where it lines out some things around stability and degradation products. Q3A-B are really the two key regulatory guidances. You've got Q2, validation of analytical procedures, and there are, if you're not aware, the USP chapters, if you're dealing with generic products or products that have been OTC, products that have been on the market a long time, these revised chapters now bring OTC and generic products into the same realm of control for impurities that you have for typical ICH or FDA type new chemical entities. So you should take a look at these new chapters, and basically they're bringing the OTCs up to date. Also, if your company goes to Brazil and markets anything in Brazil, you should be aware of these two guidances. This is actually legislation, 5315, and CP68 is a guidance for this legislation, and that really has a lot of specifics around forced degradation studies and analytical method development for those, and what you need to carry out. So it's very prescriptive and problematic if you don't do all of the conditions that they ask for. And then there's this FDA publication that really is a good one to know. I don't know the year of this, but it's fairly recent. And I'm just putting there for your own references, since you have the slides. I don't need to go through them. So impurity control strategy from ICH Q8 and Q10 is a planned set of controls derived from product and process understanding that ensure performance and product quality. So you want to control, it deals with control of the input materials, control of the processes, what goes in, what goes on, and what comes out. What goes in, what goes on, and what comes out. So your specifications, which are a combination of your limits and your analytical method, it goes together. The specifications always have a method associated with it for your impurities. And then there's a monitoring program. So the control strategy and control of what goes in, what goes on, what's going to come out, and then what's your monitoring program for those over time. Why? It's kind of an obvious. Why would we want to have impurity control strategies? Nobody wants to be taking impure drugs, just like you don't want to eat impure things, although we eat quite a variety of impurities in our daily diet. About 1 gram per day of mutagens we ingest on average. And so we control mutagens in pharmaceuticals down to in the range of 1.5 micrograms today to maybe 20 micrograms a day, in that kind of range typically. And that's the moss on the rocks on the mountain of ingested mutagens that we have. So we're probably overkill. But we want to ensure patient safety and efficacy. It's a regulatory requirement, and a quality by design approach, or a scientifically based approach for an impurity control strategy, allows you to quickly solve problems when they come up. Because if any of you with experience of having products on the market, if you got if you have on the market for 10, 15, 20 years, you are going to have some unusual things come up, and you want to be able to solve those new impurities that show up surprisingly quickly. And this kind of knowledge will help you do that. And impurities are usually considered, or you can just say they're considered critical quality attributes. This is the wording that's used in the guidances, but they are critical quality attributes of the compound. So we do impurity investigations, or investigations designed to control our impurities. I think they're the same for both process-related and degradation-related, or there's parallels to both. The idea being we want to establish a control system for process impurities that involves the processing and manufacturing conditions, as well as the methods and specifications. Same thing over here, except we're going to throw in packaging and formulation. But it's essentially parallel. There's just a different time frame in which you can iterate stability versus a step in a synthetic route. So here's my best guesstimate of the fallout rate. And the reason I'm putting this slide in here, because I think it's important to think about, if you did not have a fallout rate, in other words, if from the time a company decides to bring a compound to market in the development timeline, if you were pretty close to 100% or even 50% of success rate, you would do from the start the most thorough job you could on every aspect of impurity control strategies and every part of development, because it wouldn't be wasted money. But when you're sifting through more than 10,000 compounds, probably in the 50 to 100,000 range in discovery, and by the time you decide to do a candidate selection or whatever the term is in your company for when you're officially going to bring it, you intend to bring it into humans, it's going to be somewhere in the range of 10 to 20 to 1 in terms of you're going to have probably at least 10 to 1 or 20 to 1 fallout rate. And somewhere as development goes through this, you continue to have attrition, and maybe by phase 2B you're only in the roughly 5, 6 to 1. Maybe by phase 3 you'd like to be 1 to 1. You'd like to have pretty good confidence, but people still fail in phase 3 when the final results come out. So you've got this significant fallout rate, which means we need to think about how much money we're spending on compounds that are not going to make it. If most of the compounds you work on aren't going to make it, you better be selective in how much money you're spending. Otherwise, you're just throwing money out the window. Some key references for thinking about the development timeline I'm throwing up here for you. Not going to go through them, but it's something that you can look up for your own library. Specifications, I've already mentioned once that specifications are not just the threshold or the limit, they're the associated method. So it's right here, it's both the tests and the acceptance criteria. The acceptance criteria for impurities in drug substances and drug products are found in ICH Q3AB, which does not cover clinical development, right? Q3AB are intended for the market. So the question is, what do you do in that fallout rate during clinical development? What are the specifications? What are your thresholds for identifying, qualifying, reporting? What's your acceptance criteria for or the thresholds for mutagenic impurities is found in M7, which does cover the clinical development period. So it's a little bit easier in terms of you have to meet what it says in the guideline during clinical development. It's very clear. Because ICH Q3AB doesn't cover clinical development, the companies themselves are left to decide what kind of levels are we going to decide upon for identification, reporting, identification, qualification. Some companies just say, well, let's just do ICH Q3AB, and that's rigorous, right? But there's, and most companies wrestle through, are we going to do 2 times ICH, 3 times ICH? What's our justification? Fortunately, there's a publication by the IQ Consortium, which is innovation quality. It's like a cross-industry group of scientists that got together, and they published a series of 5 publications describing the dilemma that you face, both in analytical methodology and in specification setting. And they talk about what they propose for impurities and degradation products is essentially a 3 times ICH identification and qualification limits. So they're saying instead of 0.1% for the API, 0.3%, and for qualification, instead of, was it 1.5, it's 0.5. And so you can leverage that and point to this publication as part of your strategy. If some companies aren't comfortable going with 3 times ICH, but certainly this is a number of companies. I mean, there must be 7, 8 companies or more that publish this. And if you look at tablets and capsules, that was the API, so this would be the drug product. They're suggesting individuals unspecified of 1% and not more than 5% total. So that gives you a little bit of room that saves you money if you want to leverage that publication. They also go through, in this publication, they go through scenario of lot A and lot B and what would be the acceptability considerations. So it's very useful to look through that publication and practically think through examples, case studies that you can use for your own determination. But if you look here, again, they're at 3 times ICH, and they give you, again, 3 times ICH. So that seems to be the proposed acceptance criteria for early phase development. I think when you move to late phase development, you need to start thinking about marketed product, actual Q3AB levels. So again, it does not cover clinical development. So what about, there's GSK, Pfizer, AstraZeneca, and Janssen authors wrote a paper from a tox point of view, and they calculated what, from accepted tox criteria, what would be acceptable for a less than 6-month clinical study for a non-mutagenic impurity, and they said 5 milligrams a day or 0.7%, whichever is lower. So from a safety point of view, 3x ICH is still very conservative. I think you'll have trouble getting this approved by a regulator, but it does tell you that you can make the argument that the 3 times ICH is still significantly below any kind of a tox calculation with accepted toxicological laws and typical calculation procedures. Now let's look about mutagenic impurity thresholds. Make sure I'm not doing too slow. 15 minutes so far. So ICH M7 does cover the clinical research period. So for phase 1, phase 2, phase 3, early development, they give you actual thresholds, right? 120 micrograms per day if your clinical trial is less than 1 month, and if it's down to the most rigorous is 1.5 micrograms per day if it's greater than a 10-year exposure. And this kind of, you can see the difference there, and that applies both in clinical as well as on the market. If we think about summarizing, if we use some phase-appropriate, higher specification thresholds, it can affect what kind of the rigor that you need, analytical methods, how far you need to go down, when you need to identify. That saves you a lot of money. And here's how I would define sort of this. You can move this around a little bit. Each company is going to define this a little bit differently, but early phase development, somewhere where you've decided to pre-IND, where you've decided to bring it to market, to probably phase 2B. But you can save a lot of money doing that, a lot of lower resource burn. And what about analytical methods? So you think about drug discovery to very early phase 1. You often, many companies use screening methods that are sort of highly resolved, but sometimes generic. So something that they can have a high throughput. Hopefully they're highly resolving, but sometimes not when they're in discovery. When you get into phase 1, you want to have some kind of idea that your methods are fit for purpose, that they're going to detect the impurities that are generated and the degradation products that are generated. But as you move through to on the market, you're going to go from a limited validation set to a full validation set. Oftentimes people like to, I got to see what I have next on this. Oftentimes people like to use, as long as they can, an investigational type method with high sensitivity and resolution that's typically LCMS, so that you can quickly get molecular weight data on impurities that form, versus if you have methods that are, say, a phosphate based and then you have to create an LCMS compatible method. I think that's a perfectly good approach, but sometimes these are not very robust. So oftentimes when you get to the full validation at the end, they may be more focused on the actual impurities. They may be tailored to a specific impurity. They may be, they're going to need to be more robust, and so the whole method validation is going to need to be more complete. In this article by, again, the IQ Consortium, this is the Part 2 document. The whole publication is around the considerations with analytical method development, and they give a pretty nice table here where they show fit for purpose, or the proposed approach for method validation for early and late stage development. So you can see as you go from identification, assay, impurity tests, early and late phase, what needs to be covered and what doesn't. And I think you can leverage this article to help figure out what does it mean to have a fit for purpose method in early phase development, and how much money can that save me as an organization. So degradation products present, okay, so from the quote from ICHQ 3A and B, they put this sort of disclaimer in there because they did not want to deal with mutagenic and potentially neurotoxic impurities. They figured that's too much to handle when they were writing this in the early 90s. So they said analytical procedures should be developed for those degradation products that are suspected to be unusually potent, and they actually say impurities as well, but let's just say degradation products that are unusually potent, producing toxic or significant effects at levels not more than the identification threshold. So they're saying you might need to look way down low sometimes. I don't know how you'd suspect that if you don't know what the peak, what the identity is. So fast forward to today, ICHM 7 is in place, and that's been fully implemented since January 2016, and it really is designed to have recommendations for assessment and control of mutagenic impurities, so that the mutagenic impurities are taken down to levels where they're no longer a significant carcinogenic risk, and that would be, they've defined that as 1 in 100,000. The general focus framework of that guidance document is 1 through 9 sections. Sections 5 and Section 8 are the two sections that really deal specifically with impurities, both degradation related and process related. I'm not going to go into this, but I have it here for your reference, that for marketed products, it gives you guidance as well, and that's in Section 4 and Section 8, where it's you can look up, and I'm not a regulatory guy, so I don't want to waste our time going through that now, but just want you to know that it's there. Again, we saw this number about 120 micrograms per day for a 1 month clinical trial, exposure to the patient down to 1.5 micrograms today. So that maximum daily intake for a chronically used drug is when there is no evidence for a threshold-related mechanism. So if you have no information on the compound, you have to assume the worst. And 1.5 micrograms per day is sort of the worst-case scenario. That calculation assumes a lifetime exposure of 70 years. So already you're seeing that's conservative, because there are not many drugs that people are going to take for 70 years daily. Based on a potential cancer increase risk of 1 in 100,000. So if you compare with actual lifetime cancer risks for humans, right now it's about 41% based on this data from 2006 to 2008. So you're increasing the risk that much. So it's a very conservative approach to control of impurities. And the reason that I'm bringing this up is that some have called this the cliff of regulatory concern, because once you find out an impurity is mutagenic or potentially mutagenic, there's a 1,000-fold difference, roughly, depending on the dose, in where you have to control that to. And that control strategy is a very challenging, potentially control strategy. This is to wake you up and wake me up. So we've been talking a little bit, it's boring regulatory stuff, which I don't find exciting, but this is kind of interesting to me. The Hubble, some of you are familiar with the Hubble, hopefully most of you, the Hubble telescope. If you take a dime and hold it out at arm's length, and look at the eye of, is that Roosevelt? If you look at his eye, that's the amount of space that they were focusing in on. They focused the Hubble telescope in that amount of space right above the Big Dipper. It's a very small space, and it's very dark where you don't see anything by the naked eye. So if you look at it with your naked eye, this is what you see. Nothing, essentially, in that space. And of course, that's so small you can barely tell what's that. It's a grain of sand at an arm's length. When they looked at that, I think they looked at it for several days, and focusing in as it rotates around the Earth. They saw about 3,000 images, which turned out mostly to be galaxies, although they couldn't necessarily determine that for all of them. So what they did then was did an ultra deep field, and that same amount of space they found 10,000 galaxies in that space. So that's a lot. And so you think, I always think about, that's an example of the better the detector, the more you're going to see. And you can also put this in context of how many stars are there in the universe. If there's that many in that dark space, how many is there in a whole? They estimate it to be between, say, 0.1 to maybe 0.6 moles of stars in the universe, because we all know 6.023 times 10 to the 23rd. So if you're thinking about how many atoms of something is in a dosage form, you can start to think about that almost like stars. And the more you look, you're going to find some extraneous interference they're going to elute wherever your compound is eluting. And so the take-home message, the more sensitive your detector, the more. So that's just designed to wake you up and give you a little tidbit. But the idea of relevance being, if you're thinking of an analytical methodology where you're having to control it to either 120 micrograms, 60 micrograms, 20 micrograms, 10 micrograms, down to 1.5 microgram, as a function of daily dose, between 3 grams down to 0.3 grams, where the drug substance ICH identification thresholds for the API are here and the drug product are here. This plot will tell you what range most compounds are in. They're in the 10 to 100 milligrams, right? If you just look in this region, the green boxes are ones that you can probably do by ordinary HPLC UV detection. The yellow ones are ones that you probably can't, but you might be able to if you really worked at it. And the red ones are the scenarios where you're not going to be able to do it, so you're going to have to use a specialized technique involving derivatization or LCMS, selected ion monitoring, some kind of a more sensitive and more expensive technique. And Teasdale and Elder wrote a nice article that just came out this April that described analytical techniques and strategies for mutagenic impurities. I didn't finish the title here. I didn't put in the title. But here's one of the figures in it, method selection process. Just to tease you, to say, hey, tease you, a Teasdale article to tease you, to get you to go maybe download this article. It'd be a good one to have a reference for thinking about strategies for developing analytical methods for genotoxic or mutagenic impurities. And you've got the slide, I hope. If you look at multiple impurities, you can see this is what's the total amount of mutagenic impurities that you might be able to. We were talking about one peak. So if you think about the total, now you can get up to from 1.5 microgram for one impurity. They'll allow 5 micrograms or 30 or 60, but it doesn't change for this 1 month. So you're limited to 120. But it tells you both individual and total with a little bit more information here in the bullet points that I won't spend our time so I don't go over, because we're 27 minutes in. I'll keep you on time so you can get out of here, but we've still got more to cover, so stay awake. So options for control of impurities in the synthetic route. Some of you work on chemical process. How many of you are like organic chemists, or you work on this API synthetic route part? So this is where you live, most of you actually. Controlling impurities in the starting material. And this is a big issue right here, the starting material, the definition of the starting material and controlling the impurities. What are the thresholds you're going to put on the starting material coming in? But FDA and ICH M7 allows this to be controlled by your quality system. They're not directly mandating what kind of thresholds you need to, but they do talk about they imply 5 steps back from the drug substance. So if you have a 1, 2, 3 step to the drug substance, they're probably going to want you to look 1 or 2 steps behind into the synthesis of the starting material. Because I was in those conversations with the pharma LD kit writing ICH M7, and they went back and forth on drafts having that information in or not. So if your starting material is really close, you better think about 5 steps back. If it's 5 steps from the front, you don't have to, you're mostly just going to control it with your quality system. But if it's only 2 steps to the final from your starting material, you're going to have to think about the synthetic route and proving to them that you've done a mutagenic impurity assessment. And of course, obviously, there's the whole aspect of purge and fate and reasonably expected. What are the reasonably expected byproducts or impurities that you have to think through and justify. I'm not going to go into that too much because I think this is pretty well developed in the industry. They've been doing it for many, many years. But the guidance gives you 4 options, and this is new. These 4 options on how to control a potentially mutagenic or a mutagenic impurity that is introduced somewhere in the synthetic route. One of the ideal option is you can just argue that the chemistry is so reactive, or you know that it's going to be purged, that you do not have to get any data to prove it. So there are some things that just hydrolyze so well, and you have to make that chemical argument. There's some help out there to do that. There's a software package called Mirabilis from LASA that helps you with this whole purge fate sort of thing, and it gives estimates of risk grids for a number of reagents that are routinely used in synthetic route development. So this can help you develop your option 4 argument. Another good approach is if you can control the impurity not at the final API, but at some previous step to a level that maybe isn't all the way down to the TCC, because you know that there's some kind of purging that goes on. So maybe you control it to 100 times the level, because you can demonstrate with purge and fate that it eliminates down to below the TTC subsequent downstream. And you have to have data to show that. And then this is monitoring the impurity in the intermediate at the TTC. So if you have some synthetic route, now you're controlling it down to the TTC prior to the API. And the worst case scenario is you have to monitor the impurity in the drug substance at the TTC, because that's going to go on forever, which isn't fun. Okay, we're 31 minutes into it. We want to go 50 minutes at 19 minutes. Okay, so now we're going to think about from ICH M7, actual and potential impurities that are likely to arise. So it's not just what you know and detected. Now you got to think about potential, and not just manufacturing, but also storage. So now we're starting to move into degradation products. So you should know that any impurities that have been identified should be considered for mutagenic potential. Assessment of potential impurities likely to present should be made, and is part of the evaluation of whether or not you're going to bring them into a mutagenic risk assessment. This implies both the synthetic impurities and degradation products. So I propose, this is me proposing this, although I know that there are multiple companies that have done this approach. The question becomes, for a company, the risk is that if I identify something during my research as I'm trying to do quality by design in developing my synthetic route or my product, now I identify it, now I've got a risk. I have to do a mutagenic risk assessment because I've identified it. So I'm not going to identify anything anymore, which is the opposite of quality by design, scientific understanding. And so one of the ways that some companies have proposed to get around this, and I recommend this, is to define in your company in writing that we're going to discriminate between proposed or tentative structures based on limited characterization and confirmed structures based on a definitive structure proof. And the strategy would be you define within your company that we will focus our risk assessments on confirmed structures, not tentative. And it removes some of the risk of having electronic notebooks or people doing, you have to tell all your mass spec folks and all your analytical folks to not identify anything unless, what's your criteria for doing that. Then you can focus your criteria around what are we going to try to confirm structures for. It doesn't get you out of jail, but it makes it a more reasonable one so you can continue to let science drive your process development. Now, I'm going to claim anyway that the less difficult is the process impurities because process impurity control strategies have been underway for many, many years, and assessment for mutagenicity or genotoxicity has been underway for many years, more than a decade, more than two probably. So that is not as new as this arena. So I think that's more difficult, and it's more difficult partially because now you have to figure out, how do I iterate on a 3-year shelf life? How do I get that to be fast? And so just what defines a likely degradation product? Let's get some definitions. Let's go back to ICH Q1A R2. The definition of stress testing tells us essentially, the way I read this is that anything that you form during stress testing and identify structurally, those are likely degradation products or potential. So I say anything from stress testing is a potential degradation product because you haven't yet demonstrated that it forms under accelerated or long term. So it sort of implies in the same definition that if you can show that some degradation products don't form under long-term or accelerated, then they were only potential, they're not actual. So let's look at M7. They wrestled with that. So they provided more clear guidance and tried to define what actual impurities are. Those degradation products observed in Q3AB reporting thresholds, observed above the reporting threshold. So now they're taking it down. Actual impurities are any impurities that you see above the reporting threshold over the shelf life, with primary and secondary packaging in place. Potential degradation products would include those above the identification threshold, include those that form. So you can see what the definition is, but yet to be confirmed under long-term storage in the primary packaging. So stress testing, well, I'll get to better definitions in a second. I'll show you a scheme that outlines it a little better. So it's clear that M7 is expecting more than just looking at the actual degradation products that you see on long-term stability. And they want you to get some knowledge of relevant degradation pathways and to justify it. So there's a lot of words that were difficult to piece together to get a clear picture. So I've tried to piece it together in a couple of publications and in these slides to give you something to refer to. So in 2015, myself and people from a few different companies, GSK and AstraZeneca and somewhere else, Pfizer, got together and proposed this basically distilling down what M7 was trying to say, saying that stress testing and accelerated stability, when you those are potential degradation products, and they should be included in your SAR analysis, and obviously anything that are actual. So these are the potential degradation product feeder pool, and obviously actual degradation products go in there as well. And this flow chart will help you designing a control strategy, basically translating ICH-M7 into a visual that you can see. Here's, it's a little more complicated than that if you look at all of the words in the guidance, and so I tried to develop this to help people see what is all of the words kind of pictorially represented. So what feeds the potential degradation products that you need to evaluate for mutagenic risk? It's all of these things: accelerated stability, photostability, and knowledge of relevant degradation pathways. The knowledge of the relevant degradation pathways comes from degradation chemistry principles, which is sort of a theoretical, relevant stress testing studies, and development stability studies. So you have, if you detect a peak or a compound and identify it in stress testing studies, you still have some outs because you can determine, is that pathway relevant to your actual degradation pathways in your actual compound of interest, in your actual storage and distribution network. So I'm going to skip this, but just theoretical hypothetical means that it has not been demonstrated with data. How well can we do a theoretical prediction? There's one software package out there that does it. There really isn't any more than that anymore. It's called Zenith or Zenith, depending on if you're from Britain or the United States, which predicts chemical degradation products and pathways based on structure and conditions. How well does it work? This is the software. Gives you, right now, as of version 8 that's coming out in 2019, it has 492 reactions or transformations built into the intelligent the rules, and so it's getting to be pretty sophisticated in terms of how much chemistry it knows. When we did an assessment about when it had less than 200 rules, it was about, it looked something like this. Or Zenith would predict way more than is real, and it predicted about maybe 55 to 60% of the actuals. Now I think that same prediction, and they're starting a new study, it's probably going to predict 70, I'm guessing 75 to 80%, but there's a lot of chemistry that's still hard to predict and hard to know, and you get a lot of false predictions. So we can't just rely on our hypothetical chemistry knowledge or in silico alone. Stress testing does the best for doing that. So let's take a look at how well stress testing does. So important questions. Let's see, what am I going? Should we elucidate structures of degradation products during stress testing? There is no mandate. It's up to each individual company. I think the guidance implies it, but many people do not. So if you're going to identify structures, do you do it tentatively or confirmed? Do you do it of some subset of all the peaks you see, and how do you do that? I think here's the algorithm that I advocate, and that is you stress to the endpoint. And there's a book out on stress testing that I edited that describes the total endpoints and the conditions. Go to, shoot for 5 to 20% or so, but define major as any peak that's 10% or more of the total degradation, and greater than 20% of the 25% of the largest individual. If you do that, you'll average probably around 8 degradation products that are major on average. It'll vary from compound to compound, but you probably, and let me show you what this looks like. If you have a fake chromatogram here, API is degraded 4.46%. 0.5% is more than 10% of that, right? Because anything 4.45% would be, so you would flag these 3 peaks for identification. You would disregard these smaller peaks. That makes it clearer for you as to how you do that. I've got to figure out how to advance this. And then the second algorithm criterion would be to 25% of the largest impurity. And you see 0.5% is not 25% of 3%. So you would eliminate that. This gives you an objective way of honing in on, here's the two major peaks that we're going to focus in on the chemistry of that, and we're not going to focus in on the minor ones. It helps you to get rid of, to make it not an unduly a large amount of work. And if you do that, we've got some data that showed from data that I've mined that works pretty well, and that you form around 8.2 major degradation products, and on average in these 15 examples, there were no surprises. Now, I think you'll get surprises once in a while, and part of it is you need to make sure you're doing excipient compatibility and formulation stress testing as well. When you identify a degradation product, some people used to say to me, well, they won't be mutagenic. It's highly unlikely because it's a deactivating process. When you go through degradation, you're deactivating it. So I didn't buy that, so I decided to do some work on that. And we had a database that used to exist. Actually, it still exists in the hands of Pfizer and Lilly because they own the data. It used to be online, but it's no longer online because it took money to keep it online, and the sponsorship stopped doing that. But there were 393 drugs and about 1,200 degradation products of those drugs at the largest point. When we analyzed it, there were 356 drugs, I think, and the total number of degradants processed with 1,000. So of the 356 drugs, 35% of them had an alerting structure. In other words, they were mutagenic alerting by the general rules associated with Ashby-Tennant alerting structures. The alerts that were shared with parent are about, no, this is the degradation product. So this is the total number of degradants with alerts, 356 out of 1021, or about 35%. There's a similar number of APIs that have that. So 35% were alerting. About 20% of those had alerts shared with the parents. So then you're only really focusing on the degradants that have unique mutagenic alerts, and that's about 15%. So that tells you about 15% of the structures you identify will have a unique mutagenic alerting structure. If you look at it with a more sophisticated software package such as LeadScope or Multicase or Derek or Sarah, you're going to get down into the 5 to 8% region when we looked at it with a more sophisticated package. So now you're about of every degradation product, every 100 that you identify, maybe 5, 6 of them are going to be alerting, unique from the parent. And roughly we can expect, if you did mutagenicity Ames testing, about half of those would be alerting. And I won't tell you how we know that, but there's data to support this. So roughly 2 to 4% overall are going to be Ames positive. So you can see what kind of frequency you might run into degradation products that form mutagenic alerting structures. And there's some data here to summarize it. It's a little difficult to figure out how many times an API might have a mutagenic alerting structure, because sometimes you can have 1, 2, or 3 in the same degradation product. So it's not clear, but it's roughly in the 1 in 20 to 1 in whatever 12% is, 1 in 8, 1 in 10 sort of a range that you're going to have a confirmed Ames positive degradant that you need to evaluate for potentially forming in the drug product. If you get to that point, how do you develop a control strategy? We're at 45 minutes. I'm going fast, but I'll calm and chill get everybody back, more me than you. We're almost at the end here. But what I want to talk to you about is this is the difficult part of trying to iterate on. You have a mutagenic degradation product that is Ames positive, what's mutagenic, and now you have to try to see if it's active and controlled in the drug product. How do you assess whether it's active in your drug product, whether it forms? So the way you can do that is by a well-designed accelerated stability study. Or you can do long-term storage if you want to do a 3-year storage and see, but you're looking down there at the TTC level now. If you want to do it faster than the long-term storage, you want to focus in on 40, 75, 6 months. This is wording from the guidance itself. And you can do it in the proposed packaging with obviously a new analytical method designed for that low level. And if you want to do it faster than 6 months, you can do a kinetically equivalent short-term study in the proposed package. So how do you do a study that's kinetically equivalent to 4075 at 6 months? And I'll go into that in a second. And when you do that study, whatever that study is, I say you do 4075, 6 months, now you have to measure it. And let's say your TTC for that calculated is 10 micrograms per day, or 10 micrograms in a dosage form. You're going to have a method that's below that, right? So you're probably going to have an LOD of maybe 10%, 20% at least. It's going to be so that you can quantify in the region of concern. And if, when you do that study, the levels of that compound, here's the wording in the guidance, approach the acceptable limit. What is approach the acceptable limit? So if it's 10, does that mean if you detected it at the end of that study at 7%, at 7 micrograms, is that 70%? Is that approaching? Is 50% approaching? There's a lot of discussion that went into that, and I think you can make the case that approaching would be anything above 50% of that acceptable limit. Then you're going to have to do a little more work. I think you're in pretty good shape if it's 25% of the limit. But if it's above 50% of whatever the limit is, then I think you're approaching the acceptable limit. How do you do that study? I think I'm going to have to go through this a little fast, and I'm going to have to say you're going to have to determine what kinetically equivalent is, and you're going to need to leverage the Arrhenius equation, and you're going to either need to do it at the same relative humidity in the proposed packaging, or you're going to have to develop an ASAP type model where you'd bring in a B term, which is the sensitivity to relative humidity. But if you don't know the energy of activation, what do you do? Because the kinetically equivalent, the Arrhenius equation requires you to know that. Well, USP uses a mean kinetic temperature calculation where they assume a kinetic energy of activation, and their assumption is 19.87 kilocalories per mole. That's conservative because typically in solid state they're in the range of 25 to 30 is the more typical. So this is conservative, and you can see the effect. This is the number of days it would take to get to 4075 equivalent. If you do it at 70 degrees C, it would take you 11.1 days. If you do it at 50 degrees C, it would take you 67.8 days to be equivalent to 4075 6 months. If you know the EA, and let's say it was 30, you could do it in a very short time period. At 60 degrees, you can do it at 10 days or 2.8 days. So that's how kind of roughly how you do it. Or if you have ASAP prime, then you can do it a little more sophisticated manner. So one way you can do it is if you maintain RH at the same, it's 4075, maintain it at 75%, you don't have to worry about the B term. Then you can just use, it eliminates that term, and it's just a straight Arrhenius calculation. If you if you don't want to maintain it at the same RH, you need to develop this kind of a model and develop the B term so you can make an accurate kinetic equivalent. An example of doing some of this work was found, and this will be my case study, and I'll be done in like 3 minutes, just a little bit over. It's 49 minutes right now. So this is published in Organic Process Research and Development. This was a compound that had 2 pyridine type moieties in it, right? And the synthetic route to produce, actually here's the structure, was to take the cyanide and to hydrolyze it with hydrogen peroxide under slightly basic conditions, and it made a very fast, efficient hydrolysis to this amide. Unfortunately, when we did stress testing of that, we detected 2 N-oxides which were alerting in their structure. One of them was alerting. We decided to do Ames test on both, and both of them were actually Ames positive. So now we have degradation products from stress testing that are Ames positive that are a risk for forming in the final step of the API's hydrolysis. So what do? we developed a method that had an LOQ of 10% of the TTC level for that compound, which was assigned by toxicology of 166 ppm, and we did it down to 16 ppm. And you can see the methods here where we show at the end of the kinetically equivalent test, we don't see these N-oxide growing, although there is a little bit of growth there in the tablets. So this would be our we exposed it to both accelerated and long-term to confirm it, and it remained less than 8 ppm. And the conclusion with those degradation pathways are not active, and then we do not have to monitor that on stability over time. We're just showing that this study showed that the degradation pathways are not active, and we changed the last synthetic step so that we didn't have to worry about the synthetic route, so they got a different hydrolysis, getting rid of the hydrogen peroxide. So the take home for this part of it is that ICH-M7 is complex. I tried to graph out all of the flow for how you would do a mutagenic risk assessment for degradation products, and this is sort of the whole thing, and it's intentionally left a little jumbled because the wording is kind of jumbled. But if you really want to go through it, it does help you to kind of say, okay, what do we have to do? What are all the words put together? But the take-home method is that it's complex. And that many companies are struggling with that control strategy around mutagenic degradation-related impurities. And degradation chemistry is still immature to allow reliable in the brain or in cerebro or in silico predictions to allow you to not do stress testing studies. And this well-designed accelerated stability study is a tricky little thing, but there is guidance out there. You can expect that 2.5% to 4% of degradation-related products that are formed from APIs overall, over a large number statistically, to be Ames positive. Gives you an idea of the frequency. And you have questions that every company needs to ask that deals in this realm, is that what strategies will you use to meet M7? Should you elucidate structures? And then what's your criteria for elucidating structures, at what thresholds? And how do you decide for stress testing what to identify, and how do you prove whether a degradation product is relevant to real-world storage and distribution, and on what basis do you elucidate structures in this tentative versus confirmed? Leveraging some of those ideas can simplify this to save some money and speed up development. Back to the original take-home message, which is impurity control strategies evolve over the development timeline because there's a fallout rate. And therefore, we don't do the most thorough work from the very start because we couldn't afford it. And using chemistry to guide your scientific approach is a much better approach than just throwing techniques at it. And they're very complicated for mutagenic impurities, especially degradation related. And the reason this is important, remind ourselves that we do noble work. Some people like to badmouth the pharmaceutical industry, but there's real needs out there. And just in the last, well, I've taken up 54 minutes. There's even more than this. These are the number of deaths. It's kind of sobering. And this is just a random assortment that I got from this data here from 2015. So there are needs out there that pharmaceuticals can help lessen and help address. And I think we do noble work, and let's remind ourselves of the need for speed in order to address this. And part of the speed comes from Indianapolis 500, which is where I live, close to where Andy Miles lives. Andy likes speed, right? Okay, so that's it. That's my whole overly long 55-minute presentation, kind of fast and disjointed at times. But I hope you got the idea that these two small parts of impurity control strategies are worth thinking about to speed up development and save some resource time. So thank you.
Do we have any questions? I've got a question. I work in oncology. My name is Paul Flynn, Calero Pharmaceuticals. I work in oncology products, and with respect to M7, I think we get very lazy in oncology because we just tend to use S9 as a blanket, a simple blanket, and say that we don't need to look at genotoxicity. but it doesn't seem like that's very satisfying, and it doesn't seem like the FDA completely accepts that. Is there any risk, or do you have anything to say about that?
It certainly is clearly lined out in ICH S9, but the problem is FDA is concerned about off-label use, and they're concerned about, okay, you get it for use in a life-threatening cancer, and it's mutagenic, right? That's usually the compounds are mutagenic, or they're non-mutagenic, but they're life-saving, in a life-threatening disease state. They're worried about, okay, now we're going to use this drug for early stage and for maybe some other use that's related, and you haven't done that mutagenicity assessment for those. But it sure does seem logical to me that if the parent compound is mutagenic, controlling the impurities in there to even lower levels is providing no level of increased safety. I think S9, you want to leverage that as much as you can, but FDA is cautious and worried about people in off-label use for non-life-threatening. But if it's mutagenic, it should be pretty clear you treat them as ordinary impurities. If it's not mutagenic, but it's used for life-threatening disease, which also qualifies under S9, then it maybe is a little less clear, and maybe you have to worry about off-label use or other non-life-threatening diseases. But it is kind of muddy, unfortunately. FDA is very cautious with that, in my experience.
You had in your list of things to worry about the degradants that are observed in a photostability study.
Mm-hmm.
That made the hair on the back of my neck stand up, because I've seen a lot of compounds totally disintegrate under, or not, but you end up with a forest of peaks in your assay and related substances method that are, you know, at least from your 4075 stability data, completely irrelevant. At what point do you sort of give yourself a pass on trying to identify peaks in a forest of
I think you
degradation?
leverage the fact that you can protect the compounds from light better than you can from heat. So you might have to say, okay, if we don't want to do it, but we know it's degrading to a lot, maybe there's a protect from light, or you make sure that the packaging is such that the biggest concern is with injectables and liquids. The solid state compounds, usually if they're in a fairly light impervious, it's unusual to be a big problem. But if there is photostability is one of my hobbies, right? So photochemistry, photostability, so I am particularly interested in that. And we did line out, there's one of the articles in there, Linda Dow et al., I think it's 2013-ish or so, we lined out, I believe, how to handle the light and what to, sort of went through step by step. And if you can't find it, just send me an email. I'll send you, I think, 2 publications that might address it.
Thanks.
Well, thank you, everyone. Thanks for coming out. We really appreciate it. Thank you.